Okay, so for this third essay, we're gonna begin by talking a little bit about the blood. And so the blood, if we're putting it into a test tube, would kind of be separated into three components. And so this top part is going to be our plasma and our serum. And our serum is mainly water, but it's also made up of three things. We have fibrinogen, globulins, which are antibodies, and albumin. And then this middle layer right here is our buffy layer. And then our bottom part right here is our red blood cells. And so now we're going to talk a little bit more about how red blood cells are broken down. And so to begin, we have our reticulocytes. I just probably butchered that. And those are premature red blood cells. And those get broken down into erythrocytes. And our erythrocytes are our red blood cells. And those look like this. And then our red blood cells have an approximately 120-day lifespan until it kind of gets broken down into a little smaller thing. And this is all kind of happening in the spleen. And so we have a macrophage that comes and kind of eats the dead red blood cell here. And then there's also components of this red blood cell called a heme. And so it kind of looks like this. And we have what we call the heme group. And then if we're to blow it up, our heme has iron in the middle of it. And so when it's in the getting broken down in here, we have our heme, iron, and then there's also amino acids. But our heme moves on to our biliverdin. And our biliverdin is going to be blue, green, and this is going to be unconjugated. And then our biliverdin is going to get broken down into bilirubin, and this is yellow. And this is also we don't really need to worry about unconjugated for biliverdin here. Unconjugated. And then there's an acid called glucuronic acid. And that will make conjugated bilirubin. And this is happening in the liver as well. And then our conjugated bilirubin goes through the bile and then the intestines and gets broken down into urobilinogen. And then from here, it could go two different ways. If it goes through the kidney, we have our urobilin, which is yellow. And then if it goes through the intestines, it's stercobilin, and that is going to be brown. And then you could determine what comes out of our body depending on those colors. And so that kind of sums up blood composition and the breaking down of red blood cells. Okay, so for this next part of the essay, we're going to talk about the clotting cascade and about blood clotting. So to do that, I'm going to draw this long blood vessel. And then on this blood vessel, we have this basement membrane. And that's going to be on both sides. I'm just not going to draw it here. And then we have a platelet. And then we have these cells. And these cells are going to leak these little proteins called VWF. And so let's say that we get damage to this blood vessel. And one thing I need to mention on this basement membrane, we have a bunch of negative charges all across it. And so this VWF is going to be attracted there. And these platelets are going to go along and it's going to become activated. So this is inactivated, activated. And there's going to be all sorts of things. So we got these, and then it's going to form this platelet plug. And there's different ways we can activate these. So we have the VWFs, which are done by VWF receptors. Once they are activated, we can get our fibrinogen. And I'm going to draw what that would look like. So it's these little squiggly lines is our fibrinogen. Then we also have ADP, which come from our dense granules. And so once these can be released, we have these granules that can be released. And so we have dense granules and alpha granules. And then we have our thromboxane. So our TXA2 is our thromboxane. And so this is what is our platelet plug. And then to get our clot unclotted, it's going to be a plasminogen, which is done by TPA. And then we have our plasmin, and then our breaks down fibrin. So the next thing is, imagine, so we have this platelet plug, and I'm going to draw the clotting cascade over here, but imagine that they're happening at the same time. And so we have our factor 12 binds to the collagen, and we get 12A, meaning activated. And then that goes to factor 11, and that becomes activated. And then we have 9A, and if we have a cofactor 8, we can go back to 10. Then we got 10A, and then 10A, if we got a cofactor 5, then we get 2, then we got 2A, and then 1 and 1A. And this is our thrombane. This is our fibrinogen, and this is our fibrin strands. And so if we're to break this up right here, up here we have, so 12 binds to collagen. So this is our intrinsic pathway. And then this is our common. And then let's say we have another thing, another pathway, but instead of binding up here, we have like smooth muscle and collagen as well. And what's that has TF surrounding it, a tissue factor. Or factor 3. This can have factor 7 bind, and then that becomes factor 12A, and then we go down here to factor 10, and then 10A. And then this is our extrinsic pathway. And so that is a little bit about how blood clots and the cascade of it. And that sums up this essay.